Some of you were quite surprised yesterday when actually I said that there are research programs at the museum. It's not only about collections of dinosaurs, but it's research institutions, and we are one of the groups, one of the big groups actually, that do research on Schistosomizes. So I will talk. So it's a second parasite talk after Leishmania. And our group specifically work on schistosomes and snails, and I will go through all of it. What is it exactly? Why it's important, and how it may be interesting for you as well and for your research. So I will start a little bit about the parasite itself, then how we can find maybe a crossover between fluid mechanics modeling, and I will tell about our projects that I'm part of. And the idea of the project is basically to share a resource and find this kind of crossovers, connect different scientists. So I think it doesn't. There is a dongle. Okay. Nope. It's just this one, right? So I can go with the computer and just. No. Yeah, that's all right. No worries. Okay. So Schistosomizes, it's a parasitic disease that is affecting over 200 million people worldwide. There is some estimations that around 800 million people are at risk of Schistosomizes. So it's mostly people that live in areas where this disease is present. Okay, thank you. It's so it's caused by schistosomes, which are flatworms, and it's transmitted by freshwater snails. So that's where the snail part comes in. So I'm responsible for the freshwater snail facility at the museum. We have different snails, different species, different strains of snails, which all transmit schistosomizes. So it's mostly prevalent among children in rural communities, because that's this social group that often uses water bodies for recreation, for bathing, also for different activities. So and in Schistosoma genus, there is 23 species. 6 species are the ones that infect humans, but 3 species are the ones, the most common ones, and the ones that the most the research is on. So Schistosoma mansoni, Haematobium, and Japonicum. It's mostly present in sub-Saharan Africa, a little bit in South America, mostly coast of Brazil, and Southeast Asia, the Philippines, and a little bit in Vietnam. It's present in 74 tropical countries, and recently, a couple of years ago, there was but in modern times, first ever outbreak in Europe, in Corsica, in French island of Corsica, where a few tourists basically contracted schistosomes by swimming in kind of slow flowing rivers, lakes, and so there is a outbreak of schistosomiasis right now in Corsica. So there were actually researchers tested it and confirmed that it came from West Africa. So someone from Africa came there and spread the schistosomes. And the snails have already been there. So snails, which is the intermediate vector, they've always been in South Europe, Italy, South France, Portugal, Spain, and so on. So schistosomiasis, it's one of so-called neglected tropical diseases. It's a kind of umbrella term for different diseases that are not the big ones, the big 3 ones. And after soil-transmitted helminths, which are soil-transmitted worms, it's only the second one in terms of number of people infected. So over 200 million people. So it's a very big. It's a very big burden also for communities, since it leads to, especially in children, it stunts children development, difficulties with learning, and it's the problem of schistosomiasis. It doesn't kill you immediately, but after many years of having this disease, it leads to many problems. Could be bladder cancer, infertility with women, liver failure, and so on. So it's quite a big problem. There is a cure. It's called praziquantel. So they're anti-helminth pills. But the problem is that it doesn't prevent reinfection. There is no vaccine. There's lots of research right now trying to develop a vaccine, but so far we don't have it. So on to the cycle, which is important to understand the free living stages. So cycle was actually very interesting, and it took hundreds of years for people to understand how it works. So we have the definite host, which depending on the species, it could be different mammals or birds. We work with human schistosomizers, so it's human or primates. So eggs are being excreted from human with urine or stool, depending on the form of schistosomizers. They enter freshwater bodies like lakes or rivers. Out of the egg hatches the first stage, which is called miracidium. It's a free swimming stage, non-feeding. Its only purpose is to find the specific snail host. So it navigates to the snail. It drills through. It mostly enters through the food of the snail. Inside the snail, it transforms. There are different stages which are called sporocysts. And after some time, around a month, again, depending on the species and environmental conditions, it emerges as a second stage, which is called cercaria. And these are the ones that are dangerous to people. Again, it's a short-lived, non-feeding stage, and it sticks out and directly penetrates the host tissue. So it drills through the skin, basically. You don't have to swallow it, like many other parasites, for instance, or eat it. It drills through your skin. You can't feel it. It's painless. That's why most of the people, well, if not all, who contract schistosomizes, they are not aware of it at all. Because sometimes there are symptoms like swimmer's itch, so-called, so kind of rash or allergic reaction. Sometimes there is no. So you may have it, and you realize after a couple of weeks or months when actually it gets more serious. It's very important after coming back from endemic areas and you feel that something is wrong to immediately get tested. So we also collaborate with Hospital of Tropical Diseases in London, and they do this cyclical tests on people coming back or migrants. So there's two free swimming stages that I'm gonna talk about. So first one is the miracidium, so the one that hatches from the egg and the one that seeks for the snail. So this is the video of the egg, and the miracidium that is hatching is completely. It's due to osmotic pressure. So you can see the miracidium moving inside. The water enters the egg and causes a rupture in the shell. And the miracidium hatches. Interestingly, miracidium hatches also when it's dead. I mean, the egg bursts and then there is a dead miracidium. So it's completely, it's a physical process. It's a very, you'll see, oh, here it goes. And how fast it swims. And here it goes, yeah. So it's, yeah, it's quite interesting thing. So miracidium is a very, very interesting organism, around 150, 180 micrometers in length, depending on the species again, and it can change its shape. It's covered by 21, 22 ciliated cells with the cilia that allows it to swim. The average swimming speed around 2 millimeters per second for Schistosoma mansoni, so it's the most common species, the species that most of the research is on. But it depends on many factors, and I will talk about these factors. And we can actually influence the swimming speed, which is where the fluid mechanics comes in as well. And now it's interesting. So it's for Schistosoma mansoni is negatively geotactic, positively phototactic. So it swims towards the light in search of the snail. But it's not that simple as it is. It depends on the snail host. So for Schistosoma mansoni, the snail host is Biofalaria, and these snails tend to stay more to the surface, and therefore the miracidia actually swim more towards the surface. Whereas for other species of Schistosoma hematobium, they have different snail hosts, Bolinus, and these snails tend to stay more in murky water, and these miracidia actually are negatively phototactic, so they would swim against the light. And again, it depends. So they are really their swimming pattern depends on which snail host they want to infect. So it's quite interesting. Here you have swimming Miracidia. It's from the microscope from the top, and so you see this kind of wavy pattern. But actually, there were some people they've been trying to trace those Miracidia in 3D space, and the swimming pattern is more like a corkscrew. It's more described like this. So they're very small, very fast, and it's quite difficult to catch them. That's what we do to infect our snails, because you have to infect the snails with Miracidia. So we use the Pasteur pipette, and we put them in a cup with the snails. Here you have it's a video of a Miracidium between two glass slides. So it's trapped, it cannot swim, and you see all the cilia around, how the beating of the cilia. So that's how it looks like. There actually been quite some research on swimming patterns of Miracidia. People have been doing them since the 70s. So we just pick up few of the interesting research. So for instance, the Miracidia swimming speed is influenced by temperature. The higher the temperature, so here the temperature is going down. With the higher the temperature, the faster they move, and also temperature changes influences the rate of change of direction of Miracidia, and the light intensity as well. So the higher the light intensity, the faster the Miracidia move. But again, there are some differences depending on the species. So it's actually quite an interesting, quite complex system. But in general, swimming speed increases with temperature and light intensity. In terms of presence of snails, so for many decades, actually, people have been looking for a kind of snail-derived compound that attracts the Miracidia. There's quite a lot of research, old one and new ones. So the Miracidia are put in water with snail conditioned water. So basically they had water with snails and then used this water for Miracidia, and they were tracing the movements of this Miracidia. And they do speed up, but not immediately. So it's more complex than we thought it is. The snail conditioned water induces behavior change like chemokinesis, so random movements in proximity to the chemical. So there are differences, but they're not that straightforward. Again, it may depend on the species, may depend on exactly what the snail produces. We're not sure yet. We know that they aggregate, and what is really interesting, that the Miracidia show preference for snail conditioned water from naive means uninfected snails, as if the Miracidia would prefer snails that are not infected, because it increases their chances to develop inside the snail into the next phases. So they use water from conditioned water from snails uninfected and infected, and uninfected snails that attracted Miracidia more. So in terms of the candidates for those attractants, this is the recent interesting paper, and they identify 6 excretory-secretory proteins as candidates for Miracidia attractants. So snail in mucus, they produce a lot of different chemicals. It's very difficult to trace what exactly is the one that attracts Miracidia. But we think there could be different proteins. So they actually trace movements of Miracidia adding those different chemicals, and they were checking how this movement changes and plotting. So it's quite a big field, because it may lead to development of kind of chemosensory interference for so basically detracting Miracidia from infecting snails, so preventing infection chemically. So if we could find something and maybe may produce something, it could be our weapon against those infections. Here, maybe I'll stop this. Because I want to first, right. There is a p38 mitogen activate protein kinase, which is present on the surface of the Miracidia while it's inside the egg. When it hatches, it deactivates. And what's really interesting, there's actually research that were also done by researchers from our lab. What is very interesting, it's you can influence the speeding, you can influence the swimming of Miracidia by activating or deactivating this kinase. So for instance, when on this graph A on the top, when the inhibitor of this P38 kinase was added, the swimming speed increased of Miracidia, whereas where the anisomycin, which activates the P38, was added, it completely slowed them down. So they basically stopped swimming. The cilia stopped beating. So you can influence the swimming by different chemicals. And again, it's quite an open field. And here is the video actually that shows this whole process. How do we put it from the beginning? But okay, so maybe we'll go in a loop. So you can't see. Let me just okay. Right. So this is a control. That's how they swim. That's where the inhibitor is added. So they swim a bit faster, and that's with anisomycin, which activates. So they basically stop swimming, they stop moving, and it's a reversible process. So it doesn't kill them; it just stops cilia from beating. So it's there's quite some chemical ways to influence the swimming of Miracidia. Right. So these were Miracidia. Now we're on to Cercari, which is the second stage. So it's a stage that emerges from the snail and seeks for the definite host, so human in case of Schistosoma mansoni. So here is the SEM pictures. It's very complex structure. They're much bigger, around half a millimeter in length. And snails infected with Schistosoma mansoni shed thousands of Cercari per day. So in theory, one snail can infect thousands of people. So it's just, you need one snail really for outbreak of schistosomiasis. It depends on many factors, on environmental factors, on snail health, species, how serious is the infection, and so on. The survival of Cercari depends heavily on temperature. So it's around 16 hours, I think 20 something maximum. Again, it depends on a bit on species and temperature and other factors. They are covered by the tegument, which is this surface of flatworm, and it works as a kind of protective layer. It basically protects them from our immune system, from detecting by our immune system. It's a very interesting structure by itself. The factors that enhance shedding are most commonly are photoperiod and thermoperiod, so temperature and light. So here is the video of snail shedding Cercari in water. So what we do, we put the snail in the darkness for 1 day, sometimes 2 days, and then we put it on the lamp basically for half an hour, 1 hour, and it starts shedding. Shedding means when the parasites emerge, shedding these Cercari. They basically drill through the skin of the snail and come out, and that's how we catch them in our lab. Sometimes it kills the snail because it causes it to lose hemolymph. You can't see it on this video, but sometimes you do. Basically, snail bleeds, some of them. So it's not very, but we've been able to keep infected snails and shed them regularly for around 5 months. So they can survive for quite a long time. And imagine this in the field where you have infected snails for months, living in the lake where you use for daily activities like they do in many endemic countries for washing clothes, fishing, bathing, and so on. So it's quite a big thing. So here we have a video. It's from the top of swimming Cercari. So they are very interesting. Morphologically, they have this tail. It's called a bifurcated tail, which they basically swim tail first, head second, and they have this very interesting movement when they swim towards the surface. They stay a little bit, and then they sink to the bottom, and then again they swim up, and then they sink. So very interesting is the tail that propels them. Most of when they calculated the movement of Cercari, most of it's actually the sinking pattern, but they swim towards the surface where they have higher chances to to find the host. And the thing that that activates the Cercari is the skin-derived free fatty acids from humans, water turbulence, and shadows. So when you think about, it's actually what you have when you have a person entering water, a bit of water, water mixing, overshadowing because you have a person, and skin-derived fatty acids. So there's quite an interesting. They also have all the receptors that allow them to detect people. So here are just a few articles about. They're not from our lab, but different researchers that were trying to understand movements of Cercari, which I find it's kind of a little bit of crossover between flow modeling and our world of parasites. So in this very interesting article, I think it's from Howard University, they were trying to estimate the efficiency of how Cercari swim by using particle image velocimetry, different calculations. They even build 1 centimeter robots mimicking the movement of Cercari, and they submerge them in corn syrup, I think, to mimic the water viscosity, which is fascinating. And they were trying to replicate the movements of these parasites. So what they found, it's the Cercari, they have this 3 swimming modes. So the tail first against the gravity with fork extended. So when they go up, it's around 0.7 millimeters per second. Then there is the free sinking mode with the fork partially extended. That's around 0.1 millimeter per second. And there is this body first mode. Is when they seek for the host. So they enter the body through the head. Basically, they have enzymes that allow them to go through the skin, and then they lose the tail. So the tail actually drops out, and it is only the head that enters the human's body, and then it enters the circulatory system. It goes to the heart, goes to the lung, again to the heart, hepatic portal vein. That's where they pair up because they have the 2 sexes. There's male and female, and they end up, depending on the species, around bladder, around intestine, and that's where that's when they stay and produce eggs. So they do the whole journey inside the body. Another quite interesting article is about effects of temperatures and viscosity of water on both Miracidia and Cercarial movements. So what they found is there is a positive effect of temperature and negative of viscosity on Miracidia and Cercaria's swimming speed. The maximum distance of Miracidia and Cercaria can travel increases with temperature. So there is again hypothesis that with global warming, with increasing temperature, the distance they can travel is higher. So they can potentially infect even more people. Travel big distances in lakes. And they've also found the Reynolds number is less than 1 for both Miracidia and Cercari. So this is the swimming speed, depending on temperature for both Miracidia and Cercari. So yeah, the high-speed videography in Cercari Miracidia has gathered useful information, and I think there's a great potential for this model. And what's really interesting is the emergence time of Cercari from snail, which is highly adapted to the definite host. So here is the fantastic kind of clock graph, which shows different species of schistosome, and their different hosts. And the emergence time is really correlated with the high probability where this host may visit the water body. So for instance, Schistosoma mansoni, which is the one that infects mostly humans and primates, emerges around 11, 12 during a day, whereas the Rodeni, which is the one that infects rodents, is only in the night, because that's where the animals, the rodents, usually go to the lake. Whereas the one Maugrebowii is the one that infects antelopes and cattle, emerges twice a day, early in the morning and late in the evening. That's where the animals usually visit water bodies to drink. So it's very, very highly correlated with when the host is most likely to visit water. So here is the chronobiology of Schistosoma mansoni with the human and Rodeni on the left. And what's really interesting is that some of the schistosomes are very closely related, and you can make hybrids of them. So for instance, Mansonia and Rodeni are quite closely related, as they belong to the same phylogenetic group. And people are doing hybrids of them. And so here on the top graph, you see that the shedding pattern of Mansonia on the left, Mansonia and Rodeni. And here on the bottom graph, you see the shedding pattern of their hybrid, basically. And it's quite different. And what is interesting, this shedding pattern of hybrids persists for numerous generations. So it's an evolutionary trend, actually. So you can change this shedding pattern by making hybrids, and then it's quite interesting. It just happened that we actually got Schistosoma Rodeni in our lab very recently. We're working on establishing the full cycle. So I think, from what I know, it will be the second lab in the world right now that we know of that has this particular parasite. So a little bit on our project. I don't know how much time. Over. Okay, I'll finish 1 minute. So basically, it's a part of the project is called Schistosome Snail Resource. It's a biomedical resource, which I'm part of. It's split between two institutions, the Natural History Museum and the London School of Hygiene Tropical Medicine, with part of a group working mostly on snails. Bonnie Webster is the lead on the project, and part of the group working on rodents, because we use rodents, mice, instead of humans, obviously, to close the whole cycle. And biomedical research, why? Because there is a need for biomedical research. Most of the research is done on very few strains, very few species of schistosomes. There are there's not that many field-derived species, and we would like to have more of them for anyone who is interested, and to support different research, not only from biology, but from different fields. So our aims is to provide live material to facilitate schistosomiasis research, but not only having standard strains. Standards are the most common, but also non-standard strains, mostly from Africa, which basically means us going to the field and collect different things. So thank you very much. You're welcome to write to me if you have any questions, or to follow also our project, the SSR. Have a go and have a look at our websites or our Twitter, and we're very welcome to answer all the questions. And if you're interested in collaboration, very happy to do so.